Hi, my name is Cynthia Rather. I am the Executive Director of the Biogenics Research Chambers. Welcome to our brief video about what we do here in San Antonio, Texas. Chambers were developed to provide exposures to allergens so that new medications could be evaluated. Here's a look at the inside of our facility. There are stainless steel tubes that run along the ceiling that provide the introduction of allergen. The subjects are closely monitored by health professionals who are always in attendance. While in the chamber, subjects are seated comfortably and are permitted to bring personal electronic devices such as laptops or tablets. For extended stays in the chambers, meals are provided. We monitor our subjects' allergy symptoms by providing easy-to-complete scorecards. The scorecards are able to provide the investigator information about what types of symptoms an individual may be experiencing at precise intervals. Why are chamber studies so valuable? The answer is simple. We're able to conduct allergy studies any time of the year. Along with that, the allergy study will be more accurate. The allergen will be introduced evenly throughout the chamber to all participants so that exposure will be more consistent throughout the study group. Since the inception of the Biogenics Chambers in San Antonio, investigator and co-owner Dr. Charles Andrews has been conducting allergy research with Drs. Robert Jacobs and Dan Ramirez in this facility. Our three physicians have provided research that has evaluated medications on the basic mechanisms of nasal and ocular allergies. I'll now turn this video over to Dr. Andrews to let him tell you more about the science of exposure chambers and his own chamber experiences.
This is an appropriate time to discuss the utilization of allergen exposure chambers in clinical trials. I should offer the following disclosures. I am an owner and investigator of Biogenics Research Chamber in San Antonio, Texas. Also, I'm the Director of Clinical Research of Diagnostics Research Group. Why are chambers a solution for clinical trials? Today we'll talk about the whims of nature, the history and rationale of chambers, feasible studies, operational aspects, developments, and some predictions. This is a photo of a mountain cedar in the hill country of Texas. The mountain cedar is the only plant that pollinates during the winter in North America. For that reason, there are few confounding factors. Each male tree can produce up to 1 billion grains of pollen, and those pollen grains are easily dispersed by the wind. This figure shows grains of pollen in thousands per meter cube. On the left axis, it goes from 0 to 50,000 per cubic meter. On the x-axis, we see the dates of pollination. Although it's fairly dependable that pollen will be in sufficient quantity and concentration to cause symptoms from December 15th through mid-February, it is also evident that there's a fair amount of spread, particularly looking at the January months when counts are as high as 50,000, and the early December, late February months when looking at the mean and standard deviation, the counts can be low. The dotted line on the x-axis shows that for every day during the past 18 years, there's been at least one instance where the pollen count was 0. Therefore, with these large swings and what is arguably the most dependable of pollen seasons in the U.S., there's a lot of variability. So there are problems with natural exposure studies. As we saw in the previous figure, there are variations in timing and concentration and pollen seasons. Also, it should be mentioned that there are lifestyle differences that yield different exposures. In other words, some persons like to be outside, others during a pollen season will stay solely inside. Also, with natural exposure studies, there are always confounding environmental allergens, including house dust mite and animal danders in homes. Finally, there are variable weather conditions. A bit about the history of exposure chambers. The first multi-participant allergen facility was the Vienna Challenge Chamber built by Frederick Orach and S. Jaeger. In the Western Hemisphere, the first chamber was developed at Kingston Hospital in Ontario, Canada by Dr. James Day and professional Reginald Clark in 1981. They initially wanted to study and evaluate the effects of contaminants from formaldehyde foam insulation. 6 years later, the Kingston chamber was adapted for particulate antigen distribution for allergy studies. The concept of chamber is to mimic, The concept of chambers is to mimic nature to stimulate allergic symptoms. Stable concentrations of antigen in air over time will approximate natural exposure and stimulate symptoms of allergic rhinoconjunctivitis. Interventions for allergy can be studied in this highly controlled challenge model. There is mitigation of confounding factors. There are basins to collect tissues for each subject so that they can later be weighed. A counting platform is present in the foreground, and in the ceiling are stainless steel tubes that deliver pollen. This is a photo of the Ohio chamber in Japan. There are some differences in this chamber in that the subjects are gowned, and behind each subject is a laser counter to collect concentrations of pollen for that specific subject. If manual counters are employed, it's not necessary for the subjects to be gowned. There are several desirable design features in a chamber. First, there should be control of airflow, antigen delivery, temperature, and humidity. Confounding antigens should be eliminated, and that's done primarily by HEPA filters. The environment should be easily cleaned. There should be a reliable means of recording subjective and objective measurements. A crash cart should be available. Complete electrical backup by generator should be provided. And there should be dependable counts assuring antigen homogeneity. Here we see pollen grains through the lens of a microscope. A specific area on a slide is counted after the pollen is collected for a measured amount of time, and the concentration of pollen per cubic meter can be calculated. On the other hand, when dust mite particles are distributed, no direct counting can be done. And in fact, dust mite concentrations can be determined only after completion of a chamber exposure. Nevertheless, slides are made to estimate the concentration and compare to previous dispersal of dust mite antigen. Worldwide, multiple antigens have been validated in chambers, with validation implying both the stability of counts or concentration and also the elicitation of symptoms by the antigen. The antigens that have been validated are ragweed, Timothy grass, mountain cedar, Japanese cedar, Virginia live oak, birch, cypress, dust mite, and cat dander. When study designs are considered, chamber experiments are well suited to answer multiple questions: proof of concept, efficacy and safety, onset of action, duration of action, dose ranging, therapeutic comparison with crossover or parallel design, and for mechanisms, immunologic and epigenetic response and durability of response. Multiple interventions can be evaluated: oral, ocular, and nasal antihistamines, nasal corticosteroids, desensitization, either sub-Q, IM, patch, or slit, biologics, novel anti-inflammatory agents, as well as non-medication rinses, aerosols, and other therapies. Here is a depiction of a chamber study flow. On the left is screen and run-in, where subjects are recruited, screened, and initiated on a run-in phase. The next step, as depicted in blue, is called the priming or qualifying exposure. It's generally a 3-hour exposure with self-assessment of symptoms. Staff is monitoring those symptoms during those 3 hours. And in addition to the diaries that are kept by the subjects, objective measurements can be made. The next step, as seen in gray, is the qualifying and randomizing treatment exposure number 2. There's a 2-hour qualifying exposure, and randomized treatment is administered, followed by a 3 to 6 hour post-treatment exposure, self-assessment of symptoms, monitoring again of the staff, by staff, of subjects, and objective measurements can be taken. The study then can proceed to green, which is the crossover design exposures, where exposures 1 and 2 are repeated, but no qualification scores are necessary. During the exposures, the subjective responses are provided on a Likert scale or a visual analog scale. Objective measurements that can be taken include pulmonary function testing, nasal lavage, nasal cell collections, peak nasal inspiratory flow measurements, and slit lamp ocular exams. The nasal and ocular symptoms that are evaluated by subjects include runny nose, itchy nose, sneezing and congestion on the nasal side, and itchy eyes, watery eyes, and redness for ocular. Objective responses of instantaneous symptom scores are provided on a Likert scale, with 0 equaling no symptoms, 1 mild, 2 moderate, and 3 severe. We have also found that a visual analog scale correlates very well with a Likert scale. I think that there are certain best practices in chambers that have evolved over time. First, to reduce variability, large cohorts should be tested. Physicians should be in attendance for all chamber exposures and a crash cart available. Experienced personnel should be present in chambers throughout exposures. And pollen counts should be taken every 30 minutes from multiple stations for real-time adjustments for temporal stability. And respect should be given for circadian rhythms. With experience, trial results have taught investigators certain lessons. First, in terms of subjects. With some of the newer interventions, subjects will be committed to a trial of several years. Exclusion criteria may include prohibitions against any previous immune therapy. Therefore, large databases are required. Subjects have been exposed for as long as 8 hours at a time. Talking on cell phones is too much distraction, but you can allow subjects to text and use their computers and tablets. You cannot provide entertainment that everyone will like. Even in Texas, not everyone likes country music. There are other lessons in terms of antigens. Ragweed pollen has been the most studied of antigens in chambers. If published levels of 3,500 grains per cubic meter are maintained, major symptoms of allergic rhinoconjunctivitis are achieved. Studies have now shown in chambers that confirm that there is cross-reactivity, with examples being shard and giant ragweed and Timothy and Bermuda grass. Some pollens are more potent than others. As an example, ragweed is 3 times more potent than mountain cedar. An example of a technical challenge with some specific antigens is Virginia live oak pollen. That pollen dependably elicits symptoms, but distribution is difficult because the pollen is sticky, it tends to clump, and it's very difficult to remove from surfaces. With several antigens now, it's been shown that total symptom scores in the natural setting correlate with those in the chamber setting. The group in Germany has shown that chamber testing is just as valid in season as out of season. Single priming exposures are usually sufficient to identify which subjects should continue in a study. Several groups now have shown that there are endotypic responses to exposure. These are identified by the rapidity of response, the degree of response, persistence of response, and also the correlation with natural season. Many investigators have shown that if skin tests of 3 to 5 millimeter diameter are chosen for study entry, among that group there is no correlation with skin test size with chamber symptoms. The same is true for natural season symptoms. That cockroach sensitization mitigates symptoms from other antigens may invoke the hygiene hypothesis of allergy, asthma, that early, cleaner environments may lead to enhancement of allergy. That although non-atopic subjects have no symptoms in chamber exposure to pollen or house dust, they have inflammatory marker response. This would imply that protection could occur at the endothelial level, an observation supported by some very elegant studies by Dr. Sunil Ahuja's group at the University of Texas Health Science Center in San Antonio. Chambers have traditionally excluded persons with significant asthma from chamber studies. Inevitably, some persons are unmasked to have a degree of asthma, If those persons have bronchospasm during an exposure, several chambers have shown now that they can be treated with albuterol and returned to the chamber. Natural season studies report a moderately high incidence of allergic conjunctivitis. In chambers, the incidence of allergic conjunctivitis is high, making it a good model for ocular studies, particularly if ophthalmologic technicians are utilized to evaluate the degree of erythema. There are several recent chamber developments. In 2010, the NIH sponsored a 2-day meeting of all North American chambers to explore clinical trial design for validation. In 2015, European and North American chambers met in Zurich and Barcelona regarding standardization of chambers for studies. At the 2017 Quad AI, that's the Academy of Allergy, Asthma, and Immunology. At the 2017 Academy of Allergy, Asthma, and Immunology, Professor Ann Ellis presented a replicate study utilizing 2 allergen challenge facilities. Another recent chamber development was a study by Dr. Hendrik Nolte and Dr. Jennifer Maloney with Dr. Orach's team in Vienna that illustrates the evolution of chamber research. They were investigating sublingual immunotherapy utilizing house dust mite antigen. That study provided the first data to be published in a U.S. package insert. It also showed the improvement of symptoms of asthma with that agent. Another recent chamber development, chambers are now being utilized to look at not only pathophysiology in response to medication, but also molecular and genetic mechanisms. In this very elegant study by Dr. Sunil Ahuja and his associates, RNA-seq was utilized to show that the trait that may prevent symptoms of allergic rhinoconjunctivitis may be centered on the modulation of the epithelial barrier. I have some predictions regarding future chamber trials. The study of asthma will be a major activity. I think that mild asthmatics can be safely studied in a chamber, very similar to the technique utilized by Drs. Nolte and Moroni. Another prediction, sponsors will utilize chambers to determine specific immunologic mechanisms and genetics. Chambers will play a significant role in personalized medicine and genotype-driven therapy. My final prediction is that phase 3 trials will someday be performed in collaborating chambers. If you would like additional information about our Biogenics research chamber, please contact Cynthia G. Rather, the Executive Director.